Kalian saya tunggu dulu untuk dia record. Masih lagi tak initiate recording. Okay, Assalamualaikum. We are now live on recording. Okay, so just in case if my internet got disconnected, please do nudge me, call, WhatsApp and everything. So I'll put my phone next to me because the other day saya record tapi saya tak perasan dia dah off dah pun. I mean internet connection tu dah off. Okay, so can you see this screen that I'm sharing now? Eksperimental Chloroplast, ini boleh nampak? Need to
Boleh,
respond.
sir.
Okay, so Mani, we are on the same screen. Okay, so Bismillahirrahmanirrahim. So this is experimental chloroplast in the aquatic plant. Okay, so actually we are planning, bukan we are planning, actually we planned past time, which is last week. We planned to have this experiment face to face with you guys. So patut kita buat conduct ni face to face, tapi minggu ni tak dapat juga face to face. So we turn to plan B. So we are having this Google Meet session. So I hope you guys pay full attention on this briefing regarding this experiment, specifically chloroplast in aquatic plant. Okay, so here are the course learning objective. You guys can read this. Here are the learning outcomes. At the end of this lesson, students should be able to prepare the slide of an Elodea leaf. Yes, this experiment requires you guys to prepare a microscope slide. Okay, i.e. means later you're going to examine the internal structure of Elodea leaf, which referring to the cell membrane, cell wall, and chloroplast using microscope. Okay, so that's why it's very important. That's why we are really looking forward for this session to have it face to face with you guys. So that we can handle so that we can introduce you guys how to handle the microscope as well. We will be able to do this experiment by face to face. You guys get to hands on the experiment using the microscope, so on and so forth. Okay, so but it's okay, we're trying the best what we can do during this pandemic season. Okay, so here is the introduction. So later for your lab report, okay, you are required, again for your lab report, you are required to write down the title. Of course, the title also will carry its own mark. Dari markah dia tersendiri. The linear outcome, no, you don't have to adjust anything. Just copy and paste. I mean, you just rewrite it again, okay, in your lab report. Okay, for this lab report, we require the students to write it fully handwritten. Tak nak type. Okay, I mean, because this experiment and the other experiment, maybe another one or two more experiment, we want the student to write the lab report handwritten so that they have a good practice. We don't want them to copy and paste just for this lab report and also for next report, for practice purposes. Maksudnya untuk tujuan latihan. So kata nanti korang langsung bila masuk universiti tak pernah langsung tulis lab report guna handwritten. So we don't want that to happen. Okay, and then for the introduction, you guys, if you want to write this introduction, just pretty much copy and paste, rearrange the sentences. It is okay. Okay, for your lab report. Okay, dia nak masuk. Alright. So, for the introduction, Elodea is an excellent plant for our studies of photosynthesis and cells because the leaves are only a few cells thick. Maknanya dia nak cakap kat sini, Elodea ni adalah is very good for our study for photosynthesis and dan juga sel, sebab leaf dia, daun dia adalah sangat nipis, hanyalah setebal beberapa lapisan sel sahaja. So, they will be easy for us to observe under the microscope to look the cells and the cell parts. Mana tu dekat cell dia dan cell part dia. So this is the leaf of the Elodea. So if you may have not encountered Elodea leaf before, actually if you go to a pet shop, the one those who sells I mean aquatic plant, aquarium, fishes and also aquatic plant, you can find Elodea being sold at the shop. Okay, so this is the leaves of the Elodea, and this is the microscope view of on the leaf of the Elodea sp. Okay, So this figure 4.2 shows you the actual diagram that you should be able to observe under the light microscope. Okay, figure 4.2 shows leaf cells of an Elodea sp. These living plants cells are viewed by light microscope. Mana tak payah pun nak sampai level-level electron microscope dan bagai-bagai. Tak perlu. With a microscope, we can see the chloroplasts move around inside living cells. So here is what is interesting here. So okay, the name of this okay, this is the cell inside the Elodea leaf, so we can see each individual cell. So if you look much more detail onto these lines, actually there are dua line dekat sini, there are two lines, and another one slightly more not so visible, but there is another line over there. So it is showing you that there is a cell wall here. That's what I'm trying to point at. And beneath it, di bawah dia ada satu lagi line itu, that is the plasma membrane. So this whole rectangle here is one cell, and inside this cell, what is most visible to your eyes, what is so obvious to be visible with uh, under the light microscope, is the organelle called chloroplast. So ini adalah organelle. Okay, so dalam sel ada organelle. Nama organelle yang kita tengah tengok ni adalah chloroplast. Okay. And it is also stated here, the chloroplast move around inside this living cell. Maknanya chloroplast ni bergerak. Okay, nanti kita akan tengok the movement of this chloroplast, that what we call that what we are going to call it as a streaming chloroplast movement. Okay, nanti kita akan tengok. Okay, cytoplasmic streaming of chloroplast. Okay, chloroplast and the other living parts of cells are found near the cell wall. Dia berdekatan dengan cell wall, but not usually in the center of the cell. Maknanya bukannya dekat tengah-tengah sel ini. If you look, so ini yang dekat tengah-tengah sel pun, do remember that we are looking at this cell from the top view. So when we are looking at the top view, even though this is may appear at the center of the cell, tapi inilah yang berhampiran dengan cell wall yang bahagian atas yang kita sedang tengok lah. So actually, if I have a 3D of the cell, so most of the chloroplast, a line mostly found just beneath the cell wall, just di bawah cell wall. Since they contain chlorophyll, which are green, chloroplasts can be seen without staining and are clearly visible within living plants cell. Okay, so dia cakap dekat sini. Since they contain chlorophyll, so chlorophyll is the name of the pigment. Nama pigment yang absorb, yang buat photosynthesis tu lah. This pigment which are colored as green, dia ada dekat dalam chloroplast. So inside the organelle, inside this chloroplast organelle ni ada pigment nama dia chlorophyll, itulah yang berwarna hijau. Okay, kalau chloroplast itself tu sebenarnya dia tak berwarna hijau pun, dia colorless pun, sebab dia hanyalah a membrane sahaja. So inside the chloroplast, where you will find the chlorophyll which is green in color, that gives the chloroplast its green color. Maknanya disebabkan pigment klorofil inilah yang memberikan chloroplast itu berwarna hijau, dan kita boleh nampak dia tanpa perlu warnakan dia pun, tanpa perlu staining. However, viewing the internal structure of a chloroplast requires the magnification of an electron microscope. So dia nak cakap dekat sini, internal structure, mana kalau tengok structure di dalam chloroplast ini, kenalah pakai electron microscope. Tapi just setakat kalau tengok the structure, dia kata internal structure, mana structure di dalam chloroplast. Tapi kalau setakat nak tengok the chloroplast itself, tak perlulah sampai level electron microscope. So here are the apparatuses that you're going to require for this experiment, which is light microscope, microscope slide, the cover slip, and the material is a sprig of Elodea sp. A sprig itu satu tangkai itulah, okay, of the Elodea sp. So here are the procedures and observation. What do you need to do? First, you need to obtain an Elodea sp leaf from the main plant and place it on a clean slide. Okay, so wait. However, we are missing some apparatus over here. Actually, what should be included here is also the forceps, penyepit besi itu, and also we might need to include here is the dropper, apa ni, penitis titis air tu kan. What else? I think that's pretty much that's what's missing. And needle, jarum. Okay, to help you to guide and placing the microscope cover. Okay, so here obtain dapatkan ni daun Elodea ni daripada pokok plantu. Kat sini kena tambah using forceps, must use forceps. Later, let's say lah this experiment comes out in your practical test, you you plug the leaf using your hand, put it onto the microscope slide, straight away marks will be deducted. Saya kata marks, maksud marks itu ada S kan, mungkin bukan satu, dua atau tiga markah mungkin kau dah hilang. First, the marks will be gone for not practicing a correct laboratory skills using the forceps. Selain daripada tu ada banyak lagi lah technical skills lain yang mungkin markah kamu akan ditolak. Okay. Place one drop of fresh water onto the leaf using dropper. Using dropper. Okay, so you don't just take any glass or just dip it into water into the beaker with water and just dab it there. No, you have to use dropper. And carefully place the cover slip on top of the leaf with the help of needle dengan bantuan jarum. Okay. Actually, needle also can, toothpick is also can. I mean, we don't actually use toothpick, but technically speaking, it is possible. Maknanya secara technically bolehlah digunakan. Okay, turn the microscope to higher power and draw what you see. Oh, sorry, I missed one slide over here. I missed one point over here. Place the star under the microscope and observe under low power and medium power. So first under low power and then you adjust it to medium power. So what you're doing here is that, okay, the microscope that we have here at KMS, the lowest magnification, if I'm not mistaken, is 20 kot, bukannya 10. We don't have 10. Sorry. 10. We have 10. The lowest magnification power we have is 10. We don't have the scanning 4. Tak ada, kita have 10. So, maksud objective lens is the lens that is just on top of the stage yang korang boleh rotate tu. So, there is 10 objective power plus objective lens, ocular lens yang you look through with your eyes, there is another 10 times. So 10 times kali 10 times, actually you are looking at the magnification of 100 times. Maksudnya 100 kali pembesaran image. 10 daripada mata ocular lens darab dengan 10 objective lens, there is 100. So that is the lowest lowest magnification power that you can do with the microscope that we have here in KMS. And then you're going to turn it into medium power. Okay, medium power means if I'm mistaken is 20. After 10 is 20. Okay, so 10, 20. What else do we have? 40 and then 100 objective lens. Yeah, I'm right. So after 10 and then you turn it to 20 medium power. So there is actually 200 times, because 20 times by 10, there is 200. And then turn the microscope to high power and draw what you see. Okay, high power ni is 40. So I mean 40 objective lens. 40 darab dengan 10 times by 10, you have 400 times of actual magnification. Maknanya image tu dah dibesarkan sampai 400 kali. After 40, you have 100 objective lens. But that one we don't use in this experiment because that one requires special technique yang kita dah buat lah. Kita dah show pada last semester. That will require the oil immersion technique. So we don't do that for this experiment. So paling highest kat sini adalah 40 objective lens. There is 400 actual magnification. So once you observe under 400 magnification, barulah you are required to draw and label the chloroplast, the green objects, the cell wall, and the plasma and the cell membrane. Okay, so this will be the result for your experiment. Okay, so for these questions, we'll discuss it later, inshallah, very soon. Okay, so now I'm going to bring you to view this video. Okay, so saya ada music background lagi. Okay, so blah blah blah. So it's only 2 minutes, but it's very important for me to show you. Okay, so the cell membrane, cell wall, and chloroplast, that's what we need to be able to see. apparatus and material. This is the microscope itself. The slide down there, the cover slip over here, dropper, pin, and forceps. Okay. So here is the Elodea plant in the beaker with the water. Okay, so here's the procedure. There, you need to use a forceps, handling and obtaining the slide, the plant, the Elodea plant, and then you need to plug, koyakkan, take away the plant using forceps again. Put it on the microscope slide there. There you can see. All right, there's the Elodea plants leaf. Okay, and then place a drop of fresh water, actually, or distilled water. Okay, on top of the leaf, and then carefully place the cover slip on top of the leaf using the needle to guide the microscope cover slip. Okay, now let's observe under the microscope. Place it, this the black thing here is called as the stage. Place it on the stage, secure it with the clipper, and observe under low power. There is 10 objective lens. Okay, and then medium power, 20 objective lens, i.e. 200 times magnification. And then you see this. And finally, under high magnification power, there is 40 objective lens, i.e. 400 magnification. There, boom, you can see sekejap, you can see the micro chloroplast moving. Yes, this one. Draw the cell in the middle. And then you can see actually the chloroplast, they're still moving around. Okay, so this would be the drawing that you should do. Okay, so double line, simple line, double line shading, not mana double line tu yang garis dua kali tak payah, just one straight simple line macam tu. And this is how the level that you need to do. Cell wall, chloroplast, plasma membrane here actually down there. Okay, so that's pretty much. Okay, thank you for watching, you are welcome. Okay. All right. So just now wait, just now you guys are able to see the chloroplast moves around. Okay, wait. I'll pause it over here. Okay, you can see you can actually see this chloroplast move. So what does this movement is called? It is called cytoplasmic streaming. Actually, why do I type and Google this thing? Is that it's not like I did not know, so that it is visible on my screen. Itu sahaja. So supaya tak bila saya cakap tu, you tahulah macam mana ejaan dia. And you guys can Google it on yours by yourself as well. It is called as cytoplasmic streaming in Elodea. Okay, so you can Google over here. Okay, so there you can see, nampak tu, it's very clear. You can see that the chloroplast is moving around, and dia bergerak tu hanya berdekatan dengan cell wall. Okay, dia bukan yang gerak secara rawak, bukan yang gerak macam randomly here and there, tak. It is moving. It is moving surrounding, not surrounding, just underneath the cell wall. Okay. Cytoplasmic streaming in plants, plant cell, blah blah blah. Okay, what is cytoplasmic streaming? Is the circulation, cytoplasmic streaming circulates the chloroplast around the central vacuole in plant cell. So maksudnya cytoplasmic streaming ni menggerakkan, circulates the chloroplast sekitar central vacuole. Sebenarnya kat tengah-tengah plant cell tu ada vacuole yang sangat besar. Okay, so now we move on onto the question discussion over here. Okay, so question number 1. All right, so why would we use an aquatic plant Elodea SP for this experiment instead of a terrestrial plant? Anyone would like to try to answer? Just throw me your answer. Throw me your idea. Kenapa kita nak gunakan aquatic plant bukannya tumbuhan yang dekat darat? Kenapa? Selma, 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 do you care to share your idea or your answer? Oh, Selma.
Uh,
Agak-agak kenapa Selma?
Saya tak tahu.
Tak apa, hentam je.
Okay.
Wrong answer are not to be penalized, but to be fixed. Agak-agak kenapa Selma kita guna aquatic plant ni?
Sebab
Sebab Daun
okey.
dia tebal ke, nipis ke, sebab daun dia macam mana, ada cuticle wall ke? Agak-agak. Kenapa kita gunakan aquatic plant untuk eksperimen ini?
Sebab da aduh, daun
Tak
dah
apa.
nipis. Daun dah nipis. Tak tahu.
Yeah, sorry.
Um,
Your voice breaking up. Ah,
oh, daun
okay.
dah nipis.
Sebab daun dia nipis. You are right. Kan tadi kita dah tengok tadi kan dekat introduction ni kan. The cell wall. Ni mana tadi point dia. It is very thin. Few cells thick only. Mana tu? Ni. Okay. Few cells thick only. Alright. Okay. Thank you, Selma. Okay, and usually, no, the question. Usually for a terrestrial plant, pokok yang tinggal dekat darat ni, daun dia agak tebal sedikit. Why? Because the leaves need to have a cuticle waxy layer yang macam lapisan lilin yang licin berkilat-kilat dekat atas itu to prevent water loss, to prevent desiccation. So for aquatic plant, they don't have that. Sebab tu daun dia nipis sahaja. Okay. So jawapan dia adalah dekat sini because it is thin. Okay. Ataupun few cells thick. Where do aquatic plant get the carbon dioxide for photosynthesis? Natasha.
Yes, sir.
Natasha. Agak-agak kat mana diorang dapat CO2 ni untuk fotosintesis?
Either from the air or water.
Lagi sekali, air and water. Macam mana?
Air or water.
Air or water. Okay, air or water. Tapi ada sedikit kena fix jawapan tu. Sebab kita tahu dia ni aquatic plant. Bila aquatic plant, dia tinggal dekat dalam air. So, dalam air ada udara. Macam mana tu?
Um,
Y- y- Your answer is almost certainly right. Ingat tak last chapter yang kita belajar pasal how CO2 can be incorporated into the water, into the body of water? Macam mana CO2 boleh masuk dalam air? Diterjun ke? Dia masuk jadi macam air gas ke? So get carbonated drinks. Mula pada huruf D. It starts with letter D. Dia, dia apa? Dis. Okay, kalau bahasa Melayu lah. Mula pada huruf M. Mere. Yes, please continue.
that's an record.
Dia meres apa?
Meresap.
CO2 ni kan meresap. So betul kot tu direct translation dia actually dia dissolve lah. Larut. Bukan cakap meresap, sorry. Direct Malay translation goes wrong. So dia dissolve. Dissolve tu larut. Larut dekat dalam air. Bukan meresap. Okay. So, where do aquatic plants get CO2 for photosynthesis? For photosynthesis, dissolve from the CO2 dissolve in the water. Okay, what or actually you can fix the answer into much a nicer proper sentence lah. I'm just going you the discussion sentence. Mana ayat bincang sembang macam tu saja. What organelle in plants responsible for photosynthesis? Tadi saya tanya Selma sudah. Alia.
Yes, sir.
Ya. Apa nama organel dekat dalam plant untuk fotosintesis?
Chloroplast.
Okay, chloroplast. Okay, you got it. Sorry, macam happy yang jawapan dia betul kan. Masa kata je, aku dapat senang-senang macam happy. Yang member lain macam panas. Aku dapat senang, kau tengok nanti. Okay, Nicole, can I get your attention? Nicole?
Yes, sir.
Okay. Alright, so next question. Tadi ni dah organelle. What is the pigment found in this organelle? Maknanya chloroplast tadi tu that absorbs light to power photosynthesis. Apa jawapan ni, Nicole? Apa nama pigment dia? Tiba-tiba tak ada. Nicole?
Chlorophyll.
Yeah, chlorophyll pigment. You are certainly right. So just give you a quick heads up, maknanya apa ni? Bukan heads up, kira macam hints awal lah. Okay, chlorophyll pigment dia sebenarnya ada bermacam-macam jenis, ada banyak jenis. Dia ada chlorophyll A, chlorophyll B, fucoxanthin, xanthophyll dan sebagainya. So dah ada banyak lah chlorophyll pigment. But as for now, it is okay for you to just mention chlorophyll pigments. Okay, you are right. Okay, next question is Batrisha. No, sorry. Tadi dah hilang. Anissa tadi saya tanya. Sorry, tadi saya tanya Natasha? Saya tanya Anissa tadi kan? Hello? Natasha. Oh, Natasha.
Yes,
Well, sebelah lari ya.
yes, yes.
Okay, saya belum tanya soalan lagi kan? Okay, question number 5. If a plant were underwater, if a plant were underwater and was photosynthesizing, maknanya kalau pokok tu dekat dalam air dan di bawah air, kalau dia menjalankan proses photosynthesis, apakah gas yang would be visibly bubbling from the plant? Gas apa yang bubble-bubble keluar daripada plant tu agak-agaknya? Photosynthesize tau, bukannya respiration tau. Kau masih kat sini, Natasha.
Uh,
Ya? Yeah? Guess apa? Ya? Yeah? Hello, boleh respon? Ada line, Natasha?
sekejap saya.
Mikrofon buka. Ah, kalau kalau fotosintesis, gas apa yang dihasilkan? Gas apa yang keluar?
Oxygen.
Oxygen. Okay, walaupun dia macam agak-agak merangkak-rangkak, but you got the answer right. Okay, answer betul tu, oxygen. Tapi kalau proses tu dikatakan respiration kalau gas yang keluar adalah karbon dioksida. Sebab kita kalau fotosintesis, dia hasilkan oxygen. Okay, so last but not least, Batrisha. Batrisha, you there? Batrisha, you there? Boleh, you have a strong internet connection to respond, Batrisha? Oh, Batrisha, are you still there? Hilang terus. Oh, nah. Panggil lama hilang terus. Has left the meeting. Patut tadi panggil awal-awal. Dah tinggal berapa orang? Tinggal 6 orang je. Semua saya dah tanya dia kan. Saya terpaksa tanya Anisah lagi sekali lah. Tadi Selma baru je tanya. Oh, korang bila saya mulakan saya tanya soalan semua lari-lari. Kau siap nanti kau jumpa depan-depan. Gurau. Okay, Anisah. Anisah.
Yes, sir.
Okay, soalan ni adalah why do chloroplast move in Elodea? Kenapa chloroplast tu gerak-gerak-gerak dekat dalam Elodea? Ni jawapan dia. I mean, yeah, somewhere here. Ah, so, Anissa, are you still on my screen? Anissa, are you still on my screen?
Yes, sir.
Masih ingat belum keluar. Okay. So, okay, saya first tanyalah. What's the question just now? Okay, tadi why do chloroplast move in Elodea? So, saya nak tanya soalan pertama. Apa nama movement chloroplast tu tadi tu? Apa nama?
Cytoplasmic
Habiskan ayatnya. Apa?
Cytoplasmic streaming.
Okey, betul. So, kenapa dia nak buat ini?
Optimize exposure of light on every single chloroplast.
Ya, maksudnya kan kita tadi memang ada kat dalam air tu kan. So, kenapa nak gerak supaya hampir setiap chloroplast tu optimize apa ni? This to optimize the exposure of each chloroplast kepada cahaya matahari. So, macam tu lah dia. Okay. Actually, you also can use ayat-ayat lain lah yang maksudnya lebih kurang sama juga saya explain that. Okay, so I think that is pretty much for this session. So again, for the lab report, I require you guys to write a full lab report, handwritten, tak mau type this time, because I need you guys to have a good practice on this one. And also do remember, last but not least, after you have answered this question, you must include your references. Saya bubuh references, hujung tu ada S. So means that minimum 2, okay, but we are looking for at least 3 references. So how do you should reference your apa ni, how you should write your references, you must use the APA format. Okay, so kalau tak tahu boleh tengok dekat dalam you boleh google dekat internet macam cara tulis reference, dan macam yang saya dah ajar untuk dalam assignment sebelum ni lah. So, but you cannot refer lecture note as a reference. You also cannot use Wikipedia for a reference. Okay, macam ni. Here is what I'm going to teach you. Okay, look at this one, yeah. Cytoplasmic streaming, Wikipedia. You cannot, tak boleh. Sekejap, saya tunggu dia loading jap. You tak boleh tulis macam ni. In English Wikipedia, cytoplasmic streaming, blah blah blah. Tak boleh, because Wikipedia tidak diterima sebagai source of reference. But tetapi kita boleh ni, saya ajar korang. Can you see all these small letter, this superscript number here, 1, 1, 2, semua ni kan? So when they did this, actually, they type semua ni, this information was taken or was cited from this reference, from this journal. Macam tu. Okay, sebelum tu saya pergi juga, saya cakap lah, kenapa Wikipedia tak boleh diterima sebagai reference. Sebab bila kita buat scientific writing, Wikipedia ni tak dianggap sebagai fully formal. Kenapa? Because Wikipedia boleh diedit, boleh ditulis oleh sesiapa sahaja, even budak darjah 5 yang sudah pandai masuk ke dalam internet sekalipun. For example, tengok saya tunjuk korang, dia buka ni, and then dia boleh read, dia boleh edit. So siapa yang nak boleh edit? If dia tekan edit, budak tadika pun boleh je nak edit. Faham tak? So maksudnya, okay, yeah, basically this the coding blah blah blah. So basically sesiapa sahaja pun boleh edit and 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 alter alter the information. Sebab itulah Wikipedia tidak dianggap sebagai reference. But if you look at number 1 here, pergi turun bawah, references. These are the reference. So if you tulis ini semua, ini dianggap sebagai reference. Maksudnya, dan macam lah cara korang kena reference juga. So ini adalah nama scientist tu, ataupun nama the person who write the journal. Journal ni maksudnya yang buat finding, yang buat penyiasatan itu, yang buat ataupun buat artikel tersebut. Nama dia, bila, lepas tu tajuk jurnalnya, kalau if it is a journal, lepas tu interface focus, itulah nama jurnal penulisan saintifiknya, jilid nombor 5, maybe version 5, jilid nombor 4, ada nombor dia dan ada PM dia dan sebagainya semua. So macam tu lah, sama juga seperti yang lain. So this you can, tapi yang PMID ni tak perlulah masuk sampai kat sini. You actually just need to include up until here pun sudah cukup. This is how you should write your reference. Macam ni, Goldstein, blah, 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 sampailah sekian. Interface nama ni. Okay. So, what is this actually? Ni apa dia boleh? Ini sebenarnya journal. Nak tahu tunjuk contoh journal? Saya akan buka, open in a new tab. So, sebenarnya dia nak cakap orang ini dia dapatkan maklumat tadi tu, yang nombor 1 ni tadi, berdasarkan dia baca jurnal ini. Macam ni lah jurnal ni, ada abstract, ada banyak. Tapi nak harap korang baca banyak ni memang dah pengsan tergolek-golek dulu lah. Okay, nampak. So ini adalah semua scientific finding dengan rumus dia, formula dia, Taylor dispersion, hypex enhancement, reactive fluxes, bla bla bla, cytoplasmic streaming. Kat sinilah yang diorang tu baca dan diorang tulis, cytoplasmic streaming has been for more than 2 centuries with the over bla 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 and bla bla seterusnya. So dekat sinilah dia dapat maklumat tersebut. So sepatutnya inilah pembacaan korang. This is what we call as journal reading. Pembacaan korang yang ini kemudian korang masukkan dekat dalam korang punya reference, seolah-olah itulah reference rujukan korang yang korang dah type dekat dalam masa nak buat lab report ni. Tapi kita tahu tidaklah kan. So there is the shortcut. You can get from Wikipedia, pergi ke bawah and then ke sini. And kalau nak lagi bagus lagi sikit, baca-bacalah juga sikit. No harm pun. Bukan kau baca ni kau terus meninggal atau pengsan pun. Baca je. Ni cytoplasmic reading, streaming. So baca ni, at least you dah refer lah, walaupun you tak boleh tengok semua. Gitu. Okay, boleh kan? Alright, so I'm wishing all you guys of the best of luck again for at least for this Friday. Remember this Friday kita ada UPS. Okay. So rasanya sampai tu saja. Ada soalan setakat ni. So I will stop this recording. How do I start?
Sir, handwritten kan sir?
So come again, break up. Dia tanya balik. Alia?
Handwritten kan?
Ya betul, bertulis tangan. You are correct. So dia tulis tangan. The way how you should send to me berbagai cara. So you guys can take photo, convert into PDF, hantar ke saya dekat dalam ni nantilah. Tapi sebenarnya I'm looking forward minggu depan kalau kita boleh face to face, hantar by hand lah, macam tu. Okay, so I answered.
Okay, sir.
Okay, so I'm going to stop this recording dulu. Okay. Dia tak
So,
stop.
yang lain semua dah lost connection.
Itu dah tinggal 2-3 je kan. That's why like I'm having this nasib lah saya ada recording ni. Faham tak? Okay,